Hi, my name is Jamal Williams, and I'm from the University at Buffalo. Our study, titled Transcriptomic Analysis of Human Brains with Alzheimer's Disease, reveals the altered expression of synaptic genes linked to cognitive deficits, was conducted in the lab of SUNY Distinguished Professor Dr. Jen Yin. Our work is currently NIH funded, and we utilize BioRender for graphics that you'll see in this video abstract. Alzheimer's disease is a debilitating neurodegenerative disorder that affects approximately 45 million people worldwide and 6 million people in the U.S. Alzheimer's is most noted for patients presenting memory loss, and this could include forgetting what the day is or what the year is, forgetting friends and family, or forgetting where one's home is or where they are. And we call this cognitive decline. And one of the major questions still in Alzheimer's research is, what are the causal factors of cognitive impairment in Alzheimer's disease? And once we find those causal factors, can we translate those into therapeutic medications to treat patients for their cognitive deficits and improve their memory? In this study, we utilized large-scale transcriptomic analysis in 230 existing patient samples, including 101 from aging control individuals and 129 from patients who had been diagnosed with Alzheimer's disease postmortem. And what we found was that when looking at differential gene expression in the dorsolateral prefrontal cortex of Alzheimer's patients when compared to control groups, was that genes most related to immune response were upregulated and genes most related to synaptic function were downregulated. Now, when investigating these synaptic function genes more deeply that were downregulated, we noticed that many of them belong to functional categories involved in presynaptic function. Next, we utilized an unbiased approach in co-expression module analysis, where we created distinct network modules of genes most associated with each other by how well their expression levels correlate. Of these modules, the most prevalent was Module 1, which was functionally assigned to the SNARE complex, which is associated with presynaptic vesicles being able to dock to the presynaptic membrane and release neurotransmitters into the synapse. There were also 10 other modules that were classified, with functional groups relating to signal transduction, immune response, and apoptosis. We then validated our findings from Module 1, which dealt with a disruption in SNARE complex functionality, in a separate data set in which single-cell RNA-seq was conducted in the prefrontal cortex of AD patients. And what we found was that the same changes we originally noticed in our bulk sequencing data was also changed in excitatory neurons and inhibitory neurons of this study, confirming our results that the SNARE complex is a major mechanism disrupted in the prefrontal cortex of Alzheimer's disease. We then further examined these dysregulated synaptic genes to further understand how they may be related to cognitive decline in Alzheimer's disease. We did so by comparing our findings at the mRNA gene level to protein markers related to cognitive stability from a longitudinal study. As you can see here, proteins related to inflammation and apoptosis should be downregulated, and proteins related to synaptic function should be upregulated in cognitive stability. And what we found in our study was that those same synaptic proteins that should be increased for cognitive stability had decreased gene expression that we associated with cognitive decline, further demonstrating that synaptic deficits, especially those related to the core SNARE complex, is correlated with cognitive instability amongst Alzheimer's patients. Taken together, using various genomic techniques from various different publicly available data sets, we were able to pinpoint a specific network that we found to be associated with cognitive decline. This network was associated with presynaptic function, more specifically the core SNARE complex, which can be further explored in future studies for treatment or targets to treat cognitive decline in AD patients.